Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. So we were learning about neurobiology, where we first discussed about the history, where the different researchers they talked about their observations about nervous system and then what they concluded. So we learned about reticular theory given by Camillo Golgi and the neuron doctrine given by Santiago Ramon. Then we learned about the two important cells that are the components of our nervous system, the neurons, which are the signaling units. So in the neurons, we learned about the structural aspects of neuron as well as the types of the neurons. In the types of neurons, we learned about the categorization of the neurons based on the different properties that they exhibit, such as structural properties, like how many processes are arising from the cell body, as well as the functional properties. Then we learned about neuroglial cells, different types of neuroglial cells that are found in our central nervous system as well as peripheral nervous system. We also learned about electrical properties of neuronal membrane, the resting membrane potential and action potential. So the neurons, the membrane of the neurons, they exhibit electrical property, means they are polarized by nature when they are at rest. So that potential when the cells are at rest is called resting membrane potential, and that is usually negative compared to whatever ground level is there. So it usually ranges from minus 60 millivolt to minus 90 millivolt, and this resting membrane potential is disturbed whenever a stimulus arises, and that generates an action potential, which is positive in nature. So the action potential, it is the one which conducts the information from one part of the body to other part of the body, and that is why it is quite important property that those cells exhibit, and that makes them excitable. In today's lecture, we are going to learn about Goldman Hodgkin Katz equation, which actually gave us an overview about the electron currents that are generated across the membrane. So in last class, we learned about Nernst equation. So Nernst equation tells us about the current, the equilibrium potential of certain ion. But on the contrary, Goldman-Hodgkin-Katz equation gives us an idea about the membrane potential considering all of the ions. So it doesn't account for one ion, it accounts for all of the ions to which the membrane is permeable. So what is the equation? So let me write down the equation for you. It is Goldman Hodgkin Katz equation. In short, it is also known as Goldman equation. So the Goldman equation says that membrane potential is equal to RT by NF, where N means the charge on the ion. Okay, RT by NF into log of PK into. Okay, before writing this equation down, let me tell you that this Nernst equation, let's write Nernst equation first. So Nernst equation tells us that equilibrium potential of any ion is equal to RT by NF log of the concentration of that particular ion outside the cell upon that the concentration of that ion inside the cell. So this accounts for the concentration of ions outside as well as the inside of cell. But in the Goldman-Hodgkin-Katz equation, it considers two things. First, the concentration of ions across the membrane, like on the extracellular side as well as the cytosolic side, as well as it also considers the permeability factor, like what is relative permeability for that particular ion. So that is why in the equation both of those terms are taken into account. So and the ion concentration is not just for one ion, it considers all of the ions. So neuronal membrane is permeable to sodium, potassium, as well as chloride ion, and that is why all three ions are considered in this equation. Even though the permeability for sodium and chloride is variable and it is quite less compared to the potassium ions, but there are still some leak channels, the sodium ion leak channels present on the neuronal membrane, and that is why we also consider them as the part of the equation. So the equation is something like that. It accounts for the permeability of the ion as well as the concentration of ion outside the cell. Okay, divided by potassium ion permeability and concentration inside the cell. This multiplied by this added with all of the ions. So this is same. This continues for sodium ion as well. Its concentration outside the cell. Um, then permeability factor for sodium ion, concentration of sodium ion inside the cell. Let me just erase this. Then again, you have to consider potassium ion as chloride ion as well. But chloride ion, so it is an ion. That is why the concentration factors they will be like exactly reversed. So like permeability of chloride ion and concentration of chloride ions inside the cell divided by permeability multiplied by chloride ion concentration outside the cell. So this is Goldman-Hodgkin-Katz equation, which gives us the membrane potential by accounting all of the factors, the permeability of particular ion as well as the concentration of that particular ion across the membrane. And that is why this is a more refined version of the Nernst equation, which gives us an idea about membrane potential, whole membrane potential. Like in the last lecture, when we discussed about resting membrane potential as well as action potential, we discussed that the resting membrane potential is mainly derived through the equilibrium of potassium ions, right? And potassium ion equilibrium, the distribution of potassium ions is unequal across the membrane. Like there are more potassium ions inside the cell compared to the outside. So whenever a cell is at rest, that driving force is maintained, which creates a membrane potential that is nearly equal to the equilibrium potential of potassium ions. But it is nearly equal. It is not exactly equal to that. Why it is not exact equal? Because there are some sodium ions that are leaking through the membrane, as well as not in this case, not at the rest. Chloride ion do not slip through, but yeah, sodium ions do exchange across the membrane, which makes this resting membrane potential somewhat similar to it doesn't let it get exactly similar to the equilibrium potential of potassium ions, but it is closer to the value. It is not equal to the equilibrium potential of potassium ions, but it is closer to that. Okay, and in the same way, action potential is also it is closer to the equilibrium potential of sodium ions. It does not get equal to the sodium ion equilibrium potential, because again at this point there is some potassium efflux as well as some chloride ions. They get this chloride ion influx, they get inside the cell, and this prevents the cell to reach this certain potential. And this was the kind of drawback of Nernst equation. In Nernst equation we only consider the concentration of single ion, and that is why we can only talk about the equilibrium potential of that particular ion. But this drawback was solved with the Goldman equation, because we are considering all three ions, and that is why this gives us exact value of the membrane potential. Okay, so with this, I hope the Goldman equation property, like how we use this equation to account the steady state membrane potential, and not for like dynamic changes potential over the time, that is clear to you all. Okay, so the thing is, this is kind of clicking a snapshot of the whatever it is happening in the cell, because you can only account for the membrane potential at that particular point of time. Like this is not the cells, they are continuously working, the channels are continuously opening, and there is continuous flow of ions from one side of the membrane to other side of the membrane, right? So this is not a steady state this is not a steady state scenario. This is changing. This is transient change, right? And that is why Goldman equation again fails here. Even if that gives us a perfect value of the membrane potential, this equation doesn't account for the changes that are going on in the cell. Like for example, at some, let's say, t seconds, the concentration of potassium ions outside the cell was, let's say, some 30 millimolar, and then potassium ion concentration inside the cell was 150 millimolar. Okay, and the neuronal cell is at rest. So the thing is, at the t plus 1 second, this concentration, the values of those concentration, that is going to change. Even if it is by the small value, even if the zero point 0.1 millimolar, or let's say 0.0001 millimolar change is there, but still this is a change, right? And this change is not accounted in the Goldman equation. So Goldman equation, it only accounts for the steady state membrane potential, but not for the dynamic changes that happens in the membrane potential over time. And that is why the researchers, they came up with an analogy with the equivalent circuit model that allows us to derive the insight for both, like the magnitude of the individual currents as well as the rate at which membrane potential changes in response to certain stimulus. And this is very fantastic because with the help of a Goldman equation, or let's say Nernst equation, we were only able to predict the value of membrane potential. But by using the analogy of electrical circuits with the neural circuits, like imagining that the wiring of, like imagining the wiring of neuronal membrane is exactly like that of this electrical circuits, we are able to solve this problem. So we are able to account for the magnitude of currents, like we could only tell about the membrane potential, and through which we can drive the values of electronic currents, but they were not so accurate because we cannot account for certain ion at that point of time. And that is why the equivalent electrical circuit model, this gives us the magnitude of the ionic currents, and that too the individual ionic current. So you can measure the potassium ion current individually, the sodium ion current again, and then chloride ion current as well. This equation, all like this electrical circuit model, this also helps us to determine the rate at which membrane potential changes in response to stimuli. So for till now, with the helps of Goldman and Nernst equation, we were only able to predict the value, but now you can even calculate the rate at which that membrane potential value changes. And again, so due to this, we were able to differentiate it between the graded potential as well as action potential, because both of those depend on the stimuli. Like in the graded potential, the amount of in the intensity or the strength of the stimuli determines the depolarization part or the hyperpolarization part. So the strength of depolarization or strength of hyperpolarization, that will be directly proportional to the strength of magnitude in the case of graded potential. So it follows a graded law. But in the case of action potential, it follows all or none law. So like you need to cross a certain threshold value for action potential to generate, and once action potential is generated, no matter how strong the stimulus is going to be, or no matter for how long that stimulus is going to be, the action potential will it will propagate from one end of the neuron to another end of neuron. So it's like all or none law. If the threshold is passed, then that potential is going to that is going to travel from one end to other end all the way around. But if that threshold is not passed, it won't be generated and it won't be passed down. So it is either all or none. And that is why we call it as all or none law. So this gives, like, this electrical circuit model helped us to differentiate it between those two ideas, like the graded potential as well as the action potential. Graded potential is like a local response and it doesn't travel for the long distances, while action potential travels like from one part of the body to other part of the body through the long tubular axons. So this is again other difference. And one more difference is that the action potential, they are only depolarizing in nature, while the graded potential, they could be depolarizing as well as hyperpolarizing in nature. And the law of summation also works in graded potential. For example, let's say the stimulus is of this strength. Okay, let's say the stimulus is of this strength. In the graph below, I'm drawing the stimulus strength, and here I'm drawing the strength of the I'm drawing the strength of the graded potential. Yeah, right, graded potential. So in the stimulus strength, let's say this is the strength of stimulus, and this is the strength of stimulus over the time. Like there are multiple stimuli in the course of time t. So this is time, and this whole time course is t, and these are the continuous stimuli that are given to the neuron. This is stimulus 1, 2, and 3. So stimulus 1 is of this strength, and that is why it is going to generate a graded potential of that of this intensity. Okay, the amplitude of graded potential will be something like this. Then the next stimulus, stimulus 2, that is the strength intensity of the stimulus is high, and that is why the intensity of the graded stimulus will also be high. Then there comes the third stimulus, and it will have the higher range, right? So this is the scenario when the individual stimulus are given at the individual point of time, but they are given consecutively in this scenario. And when they are given consecutively, this graded potential that will be generated in final scenario will be something like this. So the thing is that the graded potential, they can be summed across the time, like across the membrane. But action potential, once they are generated, they passed that certain threshold value. Okay, when they pass the certain threshold value, the value of action potential, that will always be constant. They cannot be summed. They will just travel from one end to other end as it is, without decreasing the membrane potential. Like there will be jumping of the potential. So we'll discuss about saltatory jumping in next lecture. Anyway, so the action potential cannot be summed. It is all or none. While graded potential is graded. It is literally graded. So it can be summed across the membrane. So the membrane, let's learn about the electrical. So let's now learn about the electrical equivalent circuit model. Okay, equivalent circuit model. Okay, so to analyze electrical behavior of neurons quantitatively. So um, this was given, or this was this was drawn because we wanted to analyze the electrical behavior of neurons quantitatively. Okay, and we wanted to, so that for this purpose, we employed the electrical equivalent circuit, which which compared, which gave us analogy between the electrical circuits and electrical properties of neurons. For example, it gave the electrical properties of neurons, like resist, like the ion channels, the lipid bilayer, or the membrane potential, which is seen across the membrane, and they used those to have an analogy with resistors, capacitors, batteries. So the thing is that this model, the key electrical properties of neurons, like it was compared with the components like resistors. Let's say resistors in the electrical circuits. So they are kind of equivalent to ion channels. We'll discuss about how about it, like how are they. And then capacitors, which are equal to lipid bilayer. Third, batteries. Let's say there are batteries in the circuits as well. They're equivalent to the electromotive force that is driving the ion exchange, or let's say the electronic potential, the membrane potential, or equilibrium potential here. Okay, so the membrane has different properties. Capacitance. Let's discuss about capacitance first. So in your physics classes, I hope you have learned about capacitance. If you don't know about it, capacitance is the property of any object to store charge. Okay. And this property is like neurons are also the neuronal cell membrane, like the plasma membrane of neuron has the property of having capacitance, like they have capacitance, means they can store the charge. Why? Because lipid bilayer, this property arises because lipid bilayer, so plasma membrane is a lipid bilayer, right? So lipid bilayer, this works as an insulator. We already know that lipid has an insulating property, like just like wood or air. So it works as an insulator which separates two conducting environments. So here, this is lipid bilayer, this is plasma membrane of neuron, and this is going to separate two conducting environments, the extracellular matrix on one hand and the cytoplasm on other hand. So extracellular matrix and cytoplasm, both of them have some concentration of ions, right? And that is why they are conductive in nature. While this conductive environments, they are separated through the lipid bilayer, and that is why this lipid bilayer has the property to store charge, and that is why we say that it has capacitance, which is written as Cm. So the capacitance of the plasma membrane or the lipid bilayer or neurilemma, we are writing it as Cm here. Okay, so the value of capacitance is given by this formula: voltage is equal to charge upon capacitance, right? So this formula is used here. So here, this capacitance, this the capacitance of has the units farads. Okay, so this has units farads. Okay, so capacitance has the value of capacitance across the membrane of the lipid bilayer of neurons. It is 1 microfarad per centimeter square. Okay, so this is the value for the membrane lipid bilayer. So this means that so this value is quite less. So this means that very few charges they are required for establishing a significant membrane potential, like millions of ions. Okay, well, millions of ions means the charge required is quite less because ions they are carrying very less charge in some electron volts, right? So millions of ions means a very few charges required to establish a significant membrane potential, like minus 60 millivolt, that can be established only with the help of few ion channels opening, like millions of ions will drive through that few ion channels, and that will help us to establish the capacitance. So the bulk cytoplasm and extracellular matrix, they remain they remain electroneutral. This means that they do not have any property to carry out ion currents. Why? Because the separation across the membrane, that only represents a tiny fraction of total ionic charge in the solution, and that is why they are mostly electroneutral. So this is how the capacitance is given in the case of the membrane. Alongside capacitance, membrane also has resistance. Resistance, let's say resistance or let's say conductance. Both of both of them are the antagonistic properties to each other. For example, the conductance is written as G while the resistance is equal to 1 by G. So conductance is a property of an object to show the conductive property for ions. While in the case resistance means literally resistance, the potential of that object to obstruct the flow of current. So resistance and conductance, that is derived due to the ion channels in the case of neuronal membrane. Why? Because as we already discussed, lipid bilayer is separating two conductive environments. So it is acting as a perfect insulator. So it has zero conductance. Like it only has the property to store charge, but not letting the ion current flow, because lipid is it has an insulating property. So lipids they are hydrophobic, so they don't allow ions to pass through. So that is why the resistance of the lipid bilayer, the pure lipid bilayer, has the resistance infinite resistance. It is proper insulator. So the resistance is infinite while the conductance is zero. So it cannot conduct any current. But what happens is that whenever you introduce ion channels over the membrane, it allows some ions to slip through. And when some ions slip through in any direction, it gives ionic currents. And this ionic currents are generated due to the ion channels. So this shifts the infinite resistance to a finite value. And that is why we say that ion channels are the one which are giving the resisting property to that membrane. Here, the conductance is written, it is represented by the G, while resistance is represented by R. So resistance of the membrane is Rm, while the conductance of the membrane is Gm. So resistance is exactly inverse of the conductance. So that's it for the resistance. Then the third and very important property that like neuronal cells have, exactly analogous to the electrical circuits, it is the electromotive force. Again, you need some electromotive force. I will be writing it as EMF, electromotive force, which is given by batteries in the circuit. So batteries, they electromotive force, or let's say equilibrium potential. So the batteries in the circuits, like in normally physical circuits, whenever you put a battery in that circuit, the charge will flow. Okay, so like from positive end to negative end, the charge flows. So this is in the general circuits, the providing the electromotive force, this job is done by batteries. And in the case of neurons, it is done by the setting up the the equilibrium potential with due to the unequal distribution of ions across the membrane. So equilibrium potential, let's say the equilibrium potential of potassium ion, or equilibrium potential of sodium ion, or equilibrium potential of chloride ion, all of those three, they helps in setting the driving force, the electromotive force, which is going to drive the electrical currents in the circuit. So these are the properties of the neuronal membrane that are exactly analogous with the electrical circuits. So with this I have cleared the basics of the neuronal membrane, and how, like with the help of this, how I hope the scenario is quite clear, like how we can have those we can relate those two pictures. So now let's draw the equivalent circuit of neuron. Okay. So while drawing the equivalent circuit of neuron, we have to take into consideration the three ions. Okay, let's draw the equivalent circuit of neuron. So in this case, we have to consider three ions to which the membrane is permeable: sodium ions, potassium ions, and chloride ions. Okay, so we know that sodium ion channels, they are very few in number, while we have the largest largest ion channels are like mostly they conduct the potassium ions. So we have high number of potassium ion channels. Here I'm talking about leak channels, okay? And again, the chloride ion channels are very few, and they allow the movement of chloride ions rarely, only during the moment of repolarization. During depolarization, only sodium ions are involved. During repolarization, potassium as well as chloride ions are involved. Well, so Ohm's law tells us about the interplay between the ionic current, the conductance, membrane potential, means equilibrium potential in this case. So Ohm's law states that any current, it is equal to the conductance into the membrane potential difference. So in this case, when we are considering it for, like when the action potential is generated, the current there are no sodium ion current or chloride ion current, because there is no movement of sodium ions or chloride ions. And that is why there will only be potassium ion currents. So the Ohm's law says that the potassium ion current, it is equal to the conductance of, like membrane conductance for the potassium ions, and we know that this is governed by ion channels. Whatever conductance is there, like more the more ion channels are there, the more will be conductance. So the number of ion channels is directly proportional to the conductance, the conductive property of membrane. So the value is, we give the conductance and conductance will be multiplied by the potential difference between across the membrane. So what potential difference is there? So well, the resting membrane potential was Vm, and now it is reached to the equilibrium potential of potassium ions. So this, by Ohm's law, we can deduce the potassium ion current. In this way, in the same way, we can deduce sodium ion currents as well as chloride ion currents, which are nearly equal to zero in the case of depolarization. So this complete resting membrane model tells us that all those three ions, sodium, potassium, and chloride, they contribute to total equivalent circuit. So the total membrane conductance is measured in this way. So total membrane conductance for potassium ions, it is equal to the number of ion channels. So this is I whatever I told you, I'm writing it and it with an equation. So this is just equatorial depiction of that thing. So number of ion channels which are open, obviously, which are allowing the transport of potassium ions into the conductance of single ion channel. Excuse me. So total conductance for potassium ions is Nk into the conductance for a single ion. So this is for single ion channel. So whatever a single ion channel has conductance, that multiplied into total number of open channels. So this is how the conducting and the electromotive force properties tells us that electrons, like the current, the ions, they move through across the membrane and they create the ionic currents. So here I will show you a diagram which tells us about the exact wiring of the ions. So this is how the sodium chloride and potassium ions, they are laid down on the neuronal membrane. So this is how they are laid down. So here we each channel represents the conductance or the resistance. So the conductance or resistance is given by this is the symbol of resistor. Well, this is the symbol of resistor and this is the symbol of battery. So this depicts positive side and this depicts negative side. So the when we do this, when we like when we keep an analogy between the neuronal membrane and electrical circuits in our mind, it gives us this scenario, like the conductance that will be due to ion channels, and the battery, which this here it is signifying the electromotive force which is created due to unequal charge distribution, right? So this is how we laid them down, and the direction of the currents is given like this. So we already know that sodium ions are they are on numerous on the outside of the cell. That is why the electromotive force will be from out to inwards, and when the force is in this side, the current will be from the positive direction to negative direction in this okay, circuit. So when I complete the circuit, current will be from positive direction to negative direction, means in this direction, and here it will be in this direction, and here it will be in this direction. So the whatever current direction of sodium ion current is, that will be exactly opposite to those potassium as well as chloride ions. So this is the equivalent circuit. Okay, nothing, we are just putting that in parallel, because a membrane is, well, it is a parallel circuit. It's not an series circuit, because in the series current goes from one resistor to other resistor through the battery, so it travels in series. But well, in the membrane, current is traveling parallelly in this direction or in this direction, depending on which ion exchange is happening. So that is why it is a parallel circuit. So this is how the circuit will be drawn in the case of resting membrane potential. I mean, the when the membrane is at rest. So here we end with the circuit analogy. So with the help of this analogy, an equation is laid down by scientists, which is quite important one, which takes into consideration this is literally the parallel conductance equation that we use in our high school for solving the problems of the electromotive force that is given in the, like, solving the, calculating the membrane potential across the battery. So the across the battery, the membrane potential, it is given by the conductance as well as the equilibrium potential. So in this case, there are three ions. That is why we are going to consider all three of them. So conductance multiplied by equilibrium potential of that certain ion divided by conductance of that certain ion. So we are just going to sum up all of the ions. So here, let me just write the potassium ion conductance. Sodium ion conductance into equilibrium potential of sodium ions divided by the conductance of sodium ions, and then conductance of chloride ions into the electrical potential of chloride ions plus the conductance of chloride ions. So this is the general equation for parallel conducting circuit. In this case, this will be for the neuron, a neuron which is at rest. So this equation generalizes the Goldman equation into conductance-based framework. So with this, we have solved the property that instead of considering a steady-state scenario, we are actually calculating the dynamics of the electric current flow. So we can measure like the whatever membrane potential we are measuring, that will be that will be not just a snapshot of whatever is was happening over the course of time, but you can you can predict that at any point of time, even at t, even at t plus 1, you don't really need the values of the ion concentration. So with this, we are done with this lecture. In next lecture, we will learn about we'll learn about the model neuron, like how the different structural parts of a neuron, they are related to different properties. Till then, see you. Bye bye.